Okay, so very good morning to everyone gathered here. Uh, on this occasion of our ongoing distinguished lecture series to celebrate uh, our foundation month, NIBH Foundation Month, which is now reaching 1 year younger to become sweet 16, right? So I welcome you all. And before we start today's program, I will invite our director ma'am to just have few words. Please.
Yeah, thank you very much, Dr. Bapaditya Day. Greetings of the day. Good morning, everybody. Good morning, and thank you for coming, Professor Renee. So, as Dr. Bapaditya has already set a tone of this morning, really looking forward. I am really, it's a very important thing. So, it so happened I'm meeting Professor Renee for the first time, except exactly when we started floating the whole idea to get more food for thought, thinking to One Health, why not this? And I was kind of searching many people, and there comes interaction and with this everything. And then there was a paper. I was talking to Pankaj. So he's looking forward to interact with you, like all of us. So and snake venom and anti-venom and snakes coming out, and it's directly proportional to climate change. So it made more of a sense. And then when I reached out to her, she was trapped. So, thank you. So I keep it short. Thank you so much, and we all wait.
Thank you, ma'am. So on behalf of the director and the NIB fraternity, it is my great pleasure to welcome and introduce Professor Renee Maria Borges, an eminent evolutionary ecologist and professor at the Center for Ecological Sciences, Indian Institute of Science, Bangalore. Professor Borges has had an illustrious academic journey. She completed her undergraduate degree in zoology and microbiology from St. Xavier's College, Mumbai, a master degree in animal physiology from the University of Bombay, and subsequently pursued her PhD at the University of Miami, Florida, USA. After coming back to India, over the years, Professor Borges has become internationally recognized for her pioneering work in behavioral and sensory ecology, particularly focusing on plant and animal interactions. So some of the examples are like highly specialized mutualism between figs and fig wasps. And her research spans diverse areas, including conservation biology, chemical ecology, and the history and philosophy of ecological science, which have been widely profiled in many newspapers, magazine, policy papers, as well as to books. Professor Borges is a recipient of the J.C. Bose National Fellowship and is an elected fellow of the leading science academies of India, the Indian Academy of Sciences, the Indian National Science Academy, and the National Academy of Sciences, India. She has served as chairperson of DST program, Advisory Committee on Animal Sciences. And I think in this regard, she visited NIAB earlier as well. And she is one of the key member of the Western Ghat Ecology Expert Panel constituted by Government of India. Today, we are honored to hear her speak on the topic that brings together ecology, the complexity of life, and their interconnectedness. The topic, challenges of life in the natural microcosm, a small world problem. Please join me to warmly welcome Professor Renee Borges.
You can't get rid of that thing in the perfect. Okay, thank you so much, Professor Sharma, for inviting me. And am I audible?
Yes.
I am. I'm really delighted to be here. It was said that I did visit NIB in, was it 2017? I can't really remember the year, when Professor Subir Mazumdar was the founding director of this place, and he's a very good friend of mine, and we had a meeting, and it was nice for me to see NIAB beginning and soon to be Sweet 16. So we look forward to many such celebrations. So I'm a behavioral ecologist, a sensory biologist, and I work with non-model organisms. I know that all of you are also working with very difficult subjects. I don't think I could handle a buffalo or a camel, and I'm sure many of you have to do these very challenging things. I work more at the smaller scale, okay? Though I have in the past worked with animals like giant squirrels and things like that. So, understanding ecosystem functioning is really very complicated. And ecosystems, you know, even our body, in a sense, is an ecosystem. And we know how complicated our own body is. So scientists have been trying to figure out how ecosystems really work. And one of the very early ideas was developing Biosphere 1, which became Biosphere 2. And it's a little different because in Biosphere 2, it's an enclosed ecosystem. You know, the Earth as a planet, it's an open ecosystem. But over here, the idea was, if you have a closed system, you can control what's coming in, what's going out. And there were many efforts to try and understand what are the processes. If you put in a simple group of organisms, how can you monitor them? How can you study their interactions? This was a very, very expensive project, and it has kind of petered out over the years because it's just too difficult to keep it going. So, because, you know, Biosphere 2 is so expensive, people have then tried to move, you know, a little scale down. So many scientists work on what are called mesocosms. And a mesocosm essentially is, I'm pressing it hard, but it's not. Long press, long press. Okay, mesocosms are of different types. You have the famous ecotrons. The first ecotron was set up in Silwood Park in the UK about 50 years ago. But after that, we have many modifications of ecotrons. We have limnotrons, which are freshwater, big vats of you know, freshwater ecosystems. We have outdoor ponds, we have earthtrons, which why doesn't it work when I press? No.
Link to the
Sorry?
equals the number.
I think I'll just use this.
It's the number. It is small.
But I can't see it there. Doesn't matter. I'll stand here. Can you see this? Is this working? Yeah? Okay. So we have underground earth prawns, we have oceanic planktotrons, to try and simulate in a mesocosm kind of way what is happening with larger ecosystems. And of course, these are the microcosms that I'm sure all of us are very familiar with because we use them all the time. We have petri plates, flasks, where again we try to simplify, putting in a few ingredients, a few organisms, and try to see what they might be doing. So there are some very fascinating natural microcosms, and this is a classical example of a squid light organ construction. So here you have these bioluminescent vibrio bacteria that help the squid to co-construct a light organ. And this light organ gets colonized by the vibrio, and every 24 hours, the vibrio get flushed out and new vibrios are brought into the light organ. So this is a quintessential, iconic, natural microcosm. Another iconic microcosm is the pitcher plant microcosm. In India, we have only one species of pitcher plant, Nepenthes khasiana, in the Khasi Hills of Northeast India. But Southeast Asia is very rich in pitcher plants, and we've got many, many wonderful list of all of the species and the bacteria and even mosquito larvae, etc., that are endemic and that are highly specialized to live within these very, very specialized digestive external stomachs of the pitcher plant. So as I said, we have macrocosms, and now we can zoom in from the macrocosm and then come down to what I'm calling a microcosm. And this is one of the microcosms that I work on. This is the fig microcosm. This is where all the action that I will be talking about for the next maybe half an hour, all the action is taking place within this microcosm. This is the inflorescence of the fig tree, in which the wasps, and these are wasps that are carrying pollen from a tree where they were born, and they collect the pollen and then they come in, the female wasps. There is only one entrance over here, and they push their way through the single entrance, and this is where they will breed, they will pollinate, seeds are produced, and so on. So nursery pollination mutualisms are very, very fascinating because there is a give and take between the plant and the insect. Yes, the plant gives up some oviposition sites where the wasp is going to develop, the offspring of the wasps, and the insect gives the plant the benefit of pollination. So there are benefits and there are also costs to be paid by both partners. Now, nursery pollination systems are present in a variety of plants, and you can see a variety of insects also, weevils, lepidopterans, Hymenoptera. So here we have the fig wasps in the family Moraceae, the figs in the Moraceae, and the fig wasps, of course, being Hymenopterans. Now, people have, as developmental biologists, you might be interested, how on earth did this amazing inflorescence with the flowers developing internally, how did this ever develop? And it is believed that this developed from a precursor like the genus Dorstenia, where you have a flat peduncle on which the flowers are placed, and you can see the flowers over there. And then this peduncle sort of closes up, as you will see in the next slide. So here you have Dorstenia, which is the flat capitulate peduncle on which the flowers are present. Then you have Antiaropsis, which is intermediate, where the peduncle starts closing up. And finally, you have Ficus, where everything closes, just leaving one opening at the top over there. Let me see if I can get the laser pointer view. Where is the laser pointer view? Pointer options, laser pointer. I've got it, yeah. So this is that opening where you saw the wasps that were trying to come in, okay? And this is the microcosm where all the action is taking place. So the fig, the syconium, the technical term is syconium. It's a very, very ancient mutualism, about 70 to 90 million years old. And here you can see but how do I change slides? Oh, so I have two hands. Okay, ambidextrous, very good. So here you have the ostiole where the wasps will come in, and here you have the female flowers, and you have more female flowers and very, very few male flowers at the just below the ostiole, and the male flowers will develop later. So when the wasps are coming in, it's only the female flowers that are ready to receive the pollen, and much later the male flowers will develop. Oh, sorry, I'm clicking the wrong thing. So here you can see some pollinator wasps ovipositing inside the fig, and you will notice, if you can, of course, they have lost their antennae, they have lost their wings. If you remember the picture that I showed you earlier, those wasps had wings, but once they come in, the process of pushing themselves into this microcosm makes them lose their wings, lose the tips of their antennae, which means that this is an irrevocable decision. Once they have gone in, they can't come out. Okay? They can't also come out because of these bracts, which are sort of facing downwards, and they are really very tough to get through. So if a wasp has made a mistake and coming into the wrong microcosm, this wasp is doomed because the wasp cannot get back. Okay, so this has got some very serious consequences. What you also see here, the wasps are galler wasps. What this means is that they stimulate the tissue of the single flowers to develop a gall, and the black things here are the larvae of the wasps developing, and these are the seeds. So you can see, if you remember the anatomy of a flower, you have the stigma and the style, and then you have the seed developing. Here too, the wasp is developing within a flower that it has converted into a gall. Now, this is quite important to remember. So here you can see the two plant and animal partners are coexisting in this microcosm, and this makes it all very interesting, and there are many other complications that we will get to. So, as I said, this is a very unusual system because the female wasps die within the first fig that they enter. So they lay their eggs, they lose their wings, they lose their antennae. It's a very traumatic thing for these female wasps. And the male wasps that are born within this, they are wingless, they mate and they die within the microcosm. So the poor males never get to see the outside world. If you're a male, imagine you are just going to be born and then you're going to, open up the galls, mate with the females, and then you're going to die. So it's a very, very different world for females compared to the males, which makes it very, very interesting. So here, just to show you a quick life cycle. So I told you that the wasps go in, they lay their eggs, the syconium will develop, and then you will get the offspring of the wasps emerging. At this point, the males will cooperate with each other. It's quite an interesting example of cooperation between males because the males can cut an exit hole. So they cut this tunnel, and through the tunnel then the females will collect pollen from the male flowers and leave carrying the pollen and go into another microcosm. So the whole cycle can continue again. So this is really very interesting. And it's even more interesting and more intriguing because these wasps, once they leave and go out, they live only for about 24 hours, maximum 48 hours, which means that in that time, they have to find a microcosm that is available for them to get in. So this is a huge problem for them. Also, because if fig trees of the right species may be separated by many kilometers sometimes, so how do these wasps even find the right fig in which they are going to enter? And there are 850 species of figs in the world today, and each fig species has its species-specific pollinator. So the pollinator of one fig cannot enter a microcosm of another one. So there's a great amount of specificity going on. Right, so this is just a cartoon to show you the wasp without its wings and its antennae is poking its ovipositor into the uniovulate flowers to develop wasps and to develop seeds. Okay, so to continue this analogy of the microcosm, this is it's a whole world by itself, and all of us in India are very familiar with enclosed spaces like forts or castles or whatever, which are well defended. And in a fort microcosm, you have different types of occupations. You have different, individuals doing different things. You have different types of armaments to defend yourself, to protect yourself. You've got a gate which shuts when the fort needs to be protected. So keeping this analogy in mind, I'm going to now take you into details of the microcosm that I have been studying for many, many years. So this is Ficus racemosa. It's a very big tree, can grow very big. And these are the thousands of little microcosms that we study. And it's really very, very fascinating. And these are some of the students that have worked with me, postdocs over the years. There are many more, but these were what I could fit on the slide. And I'm really grateful to them. Right, so by now you should have realized that this is the gate. And this gate provides only one-way entry. So here you can see a wasp struggling to get in. There is no scope for reversing a decision to enter. Therefore, there is a very high premium on correct microcosm identification. And I've already told you that they lose their antennae, they lose their wings. So if they've made the wrong decision, they cannot revert. Okay, so this is a legitimate gate. Every fort has a gate, there's a legitimate gate. Is there another way for other organisms to enter the microcosm? And yes, there is. So here are the legitimate pollinators that have come in through the legitimate gate, but you have exploiters who never actually enter the microcosm. The only part of the exploiter that enters the microcosm is the ovipositor. So here you have one of the exploiters, which is poking its ovipositor and trying to find a target into which it's going to lay its own eggs. And these exploiters can be gallers, they can be parasitoids, which means that they parasitize the pollinator larvae or other larvae which are developing within these flowers. So there is an added layer of complication. So I've already told you about this, that you have the microcosm developing. It starts out small and then it keeps growing. And at different stages, you have different species of legitimate entrants and illegitimate exploiters. And these exploiters, as I've told you, are the ones that come from the outside and lay their eggs into the microcosm. And finally, everybody will leave altogether and they will leave the microcosm and start looking for other suitable places. So in this system of ours, we really have a nice community. We've got 7 wasp species, 4 of which are gallers. By gallers, I mean they are herbivores. They actually stimulate plant tissue to develop. And 3 of them are carnivores, which means that they develop on the larvae of these gallers. So we've got the plant, we've got the herbivore, and we've got the upper trophic level, which is the carnivore. So we've got the whole ecosystem all within this one microcosm. So what do they look like? This is the pollinator with the wings and the other ones. Here are the males of the same species. So the pollinator male, the other galler males. And you can immediately see that there is a huge sexual dimorphism. Yes, the females have the wings and the ovipositor. The males are completely different. They are neotenic. And it is only the males of the pollinator that have mandibles that are strong enough to bite through the wall and to release the females. Okay? So the males are wingless, they are born and they die within the microcosm. So we've been able to sort of almost, I know all of you worry about gestation periods in your goats and buffaloes and whatever. I think about gestation periods of my microcosm because it goes from the pre-receptive stage when the female flowers are just developing, receptive when the female flowers can receive pollen. Then you have what we call the interfloral stage where pollinators, gallers, parasitoids, everybody is developing. And then you have the stage when the wasps are leaving. And finally, within 24 hours, the microcosm becomes ready for eating. So the fruit eaters will take the seeds and disperse them. What you will also notice is that the size of the ovipositor increases with the size of the microcosm, which is obvious, right? If you are coming in when the microcosm is small, you need a small ovipositor. And when you come in when the microcosm is growing, you need a much larger ovipositor. Now, I have also said that it is only the male of the pollinator who can bite and release all the females. So, what does this mean for relative development periods of the different wasps? If a wasp comes very early, and this, if you add up all these days, it's about almost 2 months. If a wasp lays its egg very early, it has about 2 months to develop before it leaves. But if a wasp lays its egg very late in the development or the gestation period of this microcosm, it has a very much shorter time to develop. So obviously, these wasps have different strategies. They can either, develop very fast, lay very big eggs, and catch up because they all have to leave at the same time. If they wait and hang on in the microcosm, they will be eaten up by the seed consumers. The ants will come in, the monkeys, the birds, everything will come in, and then they will land up in the stomach of the consumers, which means that they have to get out at this stage. Now, this is really very interesting, and this leads us to think about all the different types of strategies that these different wasps have. And if you look at survivorship curves, this is for the adults, not for the larvae. You can see that this is the age in days for the adults. How long do the adults live? There are some wasps, I told you, the pollinators that live only 24 hours. There are the parasitoids over here, which live outside for 25 days. So from 24 hours to 25 days is the amount of variation that you have in the inhabitants of the microcosm. And this has consequences for what I will tell you about later. So in general, gallers are shorter lived than the carnivores or the parasitoids, but they vary considerably in their longevity. Now, there are other strategies also, which might be interesting to all of you. Yes.
How do you study inside? Are there any specific techniques that you use?
No, we do a lot of experiments where we can introduce combinations of things that we want to introduce, and then we have to cut them open at different time points. Yeah? So there are different egg maturation strategies also in these wasps. Some of them are pro-ovigenic, like the pollinators. All their eggs are mature when they eclose, when they come out of the pupal case. The others are synovigenic, and these gradually develop their eggs over their lifetime. And obviously, you can see that the synovigenic ones have a much longer lifespan, adult lifespan, compared to the pro-ovigenic. So you've got these two very, very different strategies. So in our community of 7 wasp species, we have been able to show that if you have any read anything about how ecological communities are stratified, there is something that we call an R and K continuum. R species are those that go very fast. They develop fast. Think of a weed or, an insect, like let's say a mosquito could be an R species, okay? Whereas something little bigger, because K species tend to be bigger also, and they will go very slow. So R is the fast strategy and K is the slow strategy. And we can partition all our wasps on this continuum, which really means that we've got a functioning ecological community within our microcosm. Now, I told you about the developmental time. We know from our experiments that the early arrivals, they actually can shorten the developmental time of the microcosm. They are very, very strong nutrient sinks and they start drawing nutrients very quickly. They form very large galls. The seeds and the pollinators are also trying to shorten the developmental time. They are also strong nutrient sinks. And the reason we think that this developmental time is shortened is because they want to reduce the time spent in the immobile, vulnerable stage. Because remember, the parasitoids or the carnivores can lay their eggs from the outside. So they want to rush through the development so that the carnivores are not attacking them. On the other hand, the later arriving parasitoids, as I told you, they're coming later, but the fig has a fixed developmental gestation time. So they are actually pushing or lengthening the developmental time to allow their progeny enough time to mature. So here we have a classic case of a trade-off between two antagonistic factors. Some species are trying to hurry up the development and some species are trying to lengthen the development. And we have an effect size of 2 to 12 days. So we have been able to demonstrate that there is really this tug of war in development that is going on. Now, to answer your question, we do a lot of manipulation, and I've had very clever students that have really been able to manipulate these microcosms very nicely. So we are able to produce microcosms that only have seeds, only have galls, or only, as usual, have seeds and galls. And we do this with a variety of different types of experimentation. So what do we find when we create these microcosms, which have only seeds, which is S, only galls, which is G, and have both the seeds and the galls of the pollinators? Here is the early developmental stage, here is the mid-developmental stage, here is the volume of the microcosm. You can see immediately that when there are galls and seeds and galls, the volume of the microcosm is much, much bigger. Seeds alone are not sufficient to make the volume of the microcosm large, which therefore means that the galls have a very, very significant effect on the volume of the microcosm. So here is how, as I've said, we can create different types of microcosms. And when we had this result that the microcosms containing galls are the ones that are much larger, we were wondering whether hormones were playing a role. So we decided to measure some phytohormones. And what we found is, so we measured indole acetic acid and transzeatin, yes? And here you can see seeds, galls, seeds and galls. Whenever there are seeds and galls, and this is early phase and mid phase, you get a spike, in this case of indole acetic acid, and similarly with transzeatin. And of course, this effect is greater in the early developmental phase than in the mid-developmental phase. So certainly, we don't know yet the source of the plant hormones. And what we are doing with bioinformatics now is trying to see whether the pollinators are themselves expressing, because some insects themselves can express plant hormones. They have genes that are analogs and they can produce these. So we are now trying to determine whether the pollinators are hijacking the plant machinery or whether they have their own machinery in order to produce these hormones. Okay, so now if we want to do a bit of accounting, because I told you there are costs and there are benefits of a mutualism, we can account and say that pollinators are much more expensive to make than seeds. So this is the mass of carbon, and this is the mass of a single female pollinator, and this is the mass of a seed. You can see that in terms of carbon, the pollinators are hugely more expensive. Similarly, in the case of nitrogen, here you have the nitrogen content of a seed and the nitrogen content of a female pollinator. So pollinators are more expensive. We also, because we are dealing with a microcosm, we wanted to look at what we call the biogeochemical niche. How are these different occupants, what kind of stoichiometric niche are they occupying? And we, measured a variety of elements, and you can see a clear sort of separation. This is the wall of the syconium, the thick wall. These are the seeds. Here is the female pollinators. Here are the male pollinators. So they are all separated in stoichiometric and biogeochemical space. They are not overlapping, which means that they have very, very specific nutrient requirements, which probably therefore allows for their coexistence. And we can do stoichiometric ratio, C is to N, C is to P, N is to P, and we can see the pollinators separating from the seeds, separating very clearly from the wall. Now, I told you that of the 850 species of figs, every species has its own pollinator. So how is this attraction of the female pollinators to the species, how is it mediated? And we know now that it is chemically mediated. I've just graphically by colors color-coded the different volatile organic compounds. You can see that the composition gets much more simplified during the development. In the fruit dispersal stage, there are only esters and alcohols which are produced, whereas in the pollination stage, there are a lot more compounds. And we've been able to identify all these compounds. And we've also looked at what we call the volatilome, which is, all the co-expression of all these compounds across the developmental stage. And we can identify clearly about 7 of these modules of compounds that co-occur, and the wasps are using these as long-distance attractants. So again, to reiterate, each of the 800 species has a unique scent blend. Many of these species occur in sympatry. What does sympatry mean? That they occur in the same place. If they occur in the same place, then how do they find each other? And this is what the acoustic community often calls the cocktail party effect. We know that many species of birds, many crickets, they all call simultaneously, cicadas. How does one species recognize the other, even though there is so much background noise? And we wanted to investigate this. So here I have graphically represented the volatilome of one wasp fig species and the volatilome of another fig species. How do the figs recognize the difference? And what we have found, and when we took fig species that are occurring in sympatry, and we take the volatiles that are produced during the pollination phase and we do a multidimensional analysis, we clearly see that every fig species has its own defined volatilome, which does not overlap with another species. So this has given us a clue, and we have continued this work, and we are now able to identify the very specific compounds or specific ratios of compounds that makes one volatilome very different from another volatilome, and therefore the figs are not able, the fig wasps are not able to make a mistake. Right, so we've also gone on to look at olfactory receptor genes because that's important to our investigation. And here too, there's something very interesting about fig wasps. So if you take honeybees, we know that there are 162 olfactory receptor genes. There are some ants which have 158 or 350. Here is our fig wasp, Ceratosol infusiceps, with only 56 olfactory receptor genes. So there has been a great amount of reduction, and that is expected when you are a specialist. You don't require to recognize, extraneous scents. You just need to recognize a small subset of them. Okay, so we've also discovered that the olfactory receptor genes have ectopic or other than antennal expression. And we were among the first to actually, and I'll show that to you in the next slide, to prove behaviorally that the ovipositor of the fig wasp can sense volatiles just like the antenna. So here you can see that there are many genes that are being expressed in the ovipositor of the fig wasp, and we had earlier, prior to this, provided behavioral evidence, which I will show you in a minute. Okay, I have 10 minutes? Yeah, okay. So we've got this microcosm, and I'm going to tell you some more stories. But now let's just take a step back and look at the outside of the microcosm, because we're still talking about the volatiles being produced by the microcosm. So we've got what we call eavesdroppers. An eavesdropper is somebody who is listening onto a conversation that they are not supposed to listen to. And in this case, it's not sound, but it is scent. So the wasps are attracted to the scent of the receptive female flowers, and these wasps are rushing in, the pollinator wasps. But these ants are also attracted to the same scent, and this is what we were able. So here you have another species of ant. And these are the wasps rushing in, pushing their way in. And here you have the ants coming and eating them up, which is obviously a very detrimental thing for the wasps. So we were able to demonstrate that the ants are actually learning the signal of the volatile organic compounds. So we showed this for two species of ants. If you remember, the B phase is the phase when the wasps are coming into the microcosm. The D phase is when the wasps are going out of the microcosm. And when we do olfactometer tests with the ants. So what is an olfactometer? It's a Y-shaped tube where we put the scent of one thing on one side and the scent of something else on the other side. And we introduce the ant into the middle, and then we allow the ant to make a choice. So it's a Y-shaped olfactometer. And we find that the ants choose the smell of the fig when the wasps are coming in and the smell of the fig when the wasps are going out. They are not interested in the smell of the fig at the other times. We further found that this response is actually a learned response. That means that the ants are not born with this innate liking for the smell. And how did we discover this? We took naive ants that had never been exposed to the scent of a fig because their nests were very, very, very far away from a fig tree. And when we take naive ants, we find that the response is 50-50, a Y-shaped olfactometer. 50% of the time you go left, 50% of the time you go right. So if it's 50-50, that means that it's a random choice. And therefore, we were able to definitively show that naive predatory ants do not respond to these odors, which means for the first time we demonstrated that ants can learn the odor of a plant. And the significance of that odor is that the ants are able to arrive at the time when the fig wasps are also present. Okay. There is another complication in this microcosm, and that is a whole community of co-evolving nematodes. And these nematodes are also very ancient, maybe about 15 to 45 million years ago. And the next few slides, I'm going to tell you some very nice stories about the nematodes. Okay, so I'm managing this is quite a task. Right, so I just want to introduce the concept of phoresis. Okay, what is phoresis? Phoresis is like hitchhiking. You hitch a ride. You don't have to fly or do anything. You don't have to pay the petrol or the diesel or the charging the electric vehicle. You just jump on and you get a free ride. So phoresis is really very, very exciting and a very interesting aspect. And I was so interested that I did write a review some years ago, but that's beside the point. The point being that nematodes are masters of phoresis, especially in our system. And in order to understand how nematodes are masters of this, I'm sure you have been exposed to the fact that nematodes have this fantastic life history stage called the Dower life history stage. It's a stage when the nematodes sense that maybe their patch of resources is getting diminished, there's too much overcrowding, and they shut down further development, okay? And this is a very, very important, crucial stage in nematode life history. So, in our system, we've got three genera of nematodes. We have a putative plant parasite, a putative animal parasite, and a putative omnivore. And these are really fascinating. The first thing we wanted to know is who are the nematodes interested in hitchhiking upon? Once again, these are species-specific nematodes. Nematodes of one fig species will not be found in another fig species. So we reasoned that, well, maybe the nematodes might choose. Whom should they choose to hitchhike on? And we did a whole variety of experiments. And these are difficult experiments because we use single nematodes that are really small. And we give them a choice of either a male pollinator or a female pollinator, a whole wasp choice. We give them volatiles to choose from. So these are agar plates, tiny, tiny 6-well or whatever, the very small cell culture agar plates in which we cast agar and then we put a layer of barium sulfate. Why do we put barium sulfate on it? So that we can track the tracks that the nematode leaves on the surface of the barium sulfate, and that's how we know where the nematode has actually gone. So we've taken cuticular hydrocarbons, we wash the surfaces of the wasps and give them that. And we give them a cafeteria choice, all the pollinators, the non-pollinators, etc., etc. And then what did we find? We found that the nematodes only choose the female pollinators. This is what we had predicted, but it was fascinating to see this actually coming out of our experiments. So once again, we do 2-choice experiments. We give the nematodes 2 choices, and you can see that in all cases, whether we give them whole wasps or volatiles or cuticular hydrocarbons, FP means female pollinator, MP means male pollinator, or female pollinator compared to a female non-pollinating wasp. And you can see that without a doubt, they are choosing the female pollinator. Can anybody tell me why are they choosing the female pollinator alone and not any of the other wasps and not the males? Why choose the female pollinator?
Most possibly because males anyway cannot go out and spread, right?
Absolutely. If they jump onto a male, the male is going to die there, right? Why not choose another female of another species? Why choose the female pollinator? Anybody? If you know how I've been telling you about pollinators and non-pollinators, can anybody tell me? The pollinators are entering the field. The non-pollinators are only poking with the ovipositor, right? So it's difficult for a nematode to navigate. The ovipositor is like a human hair. It is that thin. And the nematodes are, equally small. And these wasps are 2 millimeters in size. Okay? So this was really fascinating for us to find that the nematodes are making the choice for the female pollinator. Now the story didn't end there. Of course, we know that cuticular hydrocarbons are different and all of that. That's fine. So I had a very enterprising PhD student who decided to really go deep into this nematode problem. And here is his cartoon of what's happening. So the nematodes are jumping onto the female pollinators. These female pollinators are leaving. Then the female pollinators are coming back. And the nematodes are disembarking. So here is the embarking stage. It's like you're catching a flight. And here is disembarking. You're getting off the flight. It's exactly that analogy. You're jumping on and you're jumping off. Okay? So, we were interested, what are the nematodes using? What types of cues, especially for the live wasps, because sometimes wasps die inside. How do they know that they will jump onto a wasp that is live versus a wasp that is not live? And my student did a lot of very, very, very clever experiments with these very, very tiny nematodes using CO2 coming through a syringe, calibrating the flow, etc., in really very interesting ways. Okay, we will just skip to this. We discover that nematodes are exquisitely sensitive to CO2. And they are so sensitive to CO2 that they can tell between the CO2 being produced by a live wasp to, if you freeze kill a wasp, or there is a wasp that's naturally dead, after some time, there will be some decomposition and some CO2 released as a result of decomposition. But those quantities of CO2 are much less than the CO2 being produced by a live breathing wasp. So this student of mine was really very, very capable and able to do very clever experiments, was able to deliver these very tiny amounts of carbon dioxide, calibrated amounts. And this is the amount of CO2 released by a wasp per hour. He was able to calibrate the release and found that the nematodes can even pick up the CO2 released by a single wasp and be attracted towards it. Okay, so this is really very smart and clever experiment. My student also developed flight assays, and I'll show you some flight assays in a little bit, but these are some results. He infested the nematodes onto experimental wasps in different number combinations. So here is control where there are no nematodes, 10, 20, 30, 10, 20, 30 of the plant parasite and the animal parasite. And you can see that when the wasp is carrying 30 nematodes on its body, its flight duration drops drastically, okay? If it's carrying a plant parasite, it's not so bad, but the animal parasite is dropping drastically. He also calibrated predation risk. So when the wasps are coming back and the disembarkation phase is happening, those wasps that are carrying more nematodes take much longer to get inside the osteo. What is the consequence? If they take long to get inside the ostiole, what's going to happen to them? From what I have shown you, what will happen to them? The ants are going to pick them up, right? So that means the predation risk is higher. So here again, you can see that if you are having more nematodes, your predation risk is higher. Lifetime reproductive success of the wasp, the number of offspring they produced, also comes down with more load of nematodes. The growth of the microcosm, because I've already shown you that the galls of the pollinators contribute to the growth of the microcosm. So if you have fewer offspring, your microcosm will not grow very much. So nematodes infestation also reduces microcosm growth. Okay, so if there are so many bad effects of the nematodes, how have the nematodes remained in the system for so many millions of years? Why haven't the nematodes driven the wasp and the figs to extinction? Because this is a very important question. So it's important, therefore, to look at real natural numbers, because in our experiments, we put 10, 20, 30 nematodes. We loaded the wasps. But what is really happening in the real world? So in the real world, these are the numbers of nematodes. You can see that very few wasps have 30 nematodes on them. Most of them will have between 0 and 5. This is in the jumping on stage. And then when you come into the jumping off stage, which is the arriving stage, you see that most of them have again between 0 and 5. What does this tell us? This tells us that there is a very strong natural selection for fewer wasp riders. What does this mean? The nematodes are the riders. They are riding on the wasps. So we don't know, and this is a big question that we have to address. How are the wasps regulating the number of nematodes on them? Can they regulate, or maybe they don't regulate? If they are not able to regulate, then the system is likely to crash. So these are some very important insights. Okay.
What is their role in peak development? Like, wasp has a role. What is the role of this department?
We don't know yet. We think they are just parasites, but they are kind of benign in the sense that if they become aggressive and overload the system is likely to crash. So they are there, but they are not, perturbing the system so much.
The NPC has a wasp and a nematode.
Yes, yes, yes. So this, if you remember, I showed you the slide of all the armaments. So this just shows you again the different ovipositors of the different members of the community. We've also done some micro piconewtons. So here we have measured the force required by this tiny hair-like ovipositor to actually penetrate into the wall of the syconium. And you can see that as the development proceeds, it becomes tougher to poke through the wall. And therefore, you need a stronger ovipositor which will be able to do the job. Okay, I'm going to start wrapping up now. I told you that we have transcriptomic evidence that the ovipositor is an olfactory organ. But before we got the transcriptomic evidence, we had behavioral evidence because we modified an electroantennogram which entomologists use to see whether the antenna is responsive to volatile compounds. So we modified that and developed an electro-ovipositogram. So we put the ovipositor between electrodes, completed the circuit, and then fitting it up to a GC-FID trace, we are able to show that the ovipositor actually fires and has a spike similar to what the antenna would do to particular volatile compounds. So we were the first really to demonstrate in a behavioral way that the ovipositor of insects is an olfactory organ. Okay, so I'm going to skip some of these stories and end with this last story. I told you that fig wasps that are born in one microcosm have to find another microcosm to complete the whole cycle. Now, the wasps are also blown in by the wind. They do fly, but a lot of times they also use the wind in order to get to their destination. But once they smell the fig, they have to come down from the, and they need active flight, therefore, to locate these microcosms. Okay, so we did these interesting and difficult experiments. So we measured what is called tethered wasp flights, where we tether the tiny wasp, 2 millimeters, onto the tip of a syringe. And then we have what is called an optical tachometer. And what this does is, it cuts the beam, a laser beam, so that every flap of the wing will cut a laser beam, and that will then, the cutting of the laser beam will be stored and acquired in a computer. Yeah. So this is just to show you the videography of how this process works. So this is how we measure flight duration. A freshly emerged wasp, how much fuel does it have in order to fly up to exhaustion, so it doesn't fly anymore? We also measured the lipids, so the fuel stores for the flight. We measured metabolic rates in a CO2 analyzer. And what did we find? So if you remember, I told you that there were fast wasps and slow wasps in a continuum. And we see that with flight duration, we see that the slow wasps have very little flight duration. The fast wasps can do a lot more. The metabolic rates are also lower. And the slow wasps also have lower lipid content. So what is the consequence of all of this? What does this mean? Oops, sorry. Somebody could say that, okay, you've got results from one fixed species. Does this really apply to all the communities of other fixed species? And in order to prove that it does, we took another fixed species, Ficus hispida, where the trees are much closer to each other, compared to our model fig, where the trees are much more separated. You would predict that if the trees are closer, you won't need so much metabolic fuel, you won't need so much flight duration, because you will find a suitable tree close by, compared to another species where the trees are much further separated. And that's exactly what we found. This is Ficus hispida, this is Ficus racemosa. The flight duration of all the wasps of hispida is much lower than the flight duration of racemosa. Lipid stores are much lower compared to racemosa. Metabolic rates are much lower compared to Ficus racemosa. So it shows that the communities of these wasps have also adapted the amount of fuel, the amount of flights that they need to do based on the resources available in the environment. Finally, we tried to model this theoretically, and we have been able to show that if you want to keep this whole community, and this is conservation biology, how would you be able to keep this community intact over many, many years? And we were able to determine by modeling that this entire wasp community requires a larger number of trees. So as trees start being pulled out of the environment, you will not get the wasps finding the right resource at the right time, and the community will start to collapse. Okay, so I have shown you many, many interactions in this microcosm. I hope that I've been able to. Okay, you might ask me, haven't really said anything about changing environments other than removing trees, but we know that these wasps are very sensitive to temperature increases. So as Hyderabad is getting hotter, Bangalore is getting hotter, everything is getting hotter. This is affecting the mortality of the wasps. The wasps are dying in large numbers. Bees are dying, pollinators are dying. Insectivorous birds that are feeding on insects are less in number. So if you affect the insects, you affect the higher trophic levels also. So I think I'm going to end here by saying that there are many species of figs with lots of different microcosms which have many interesting stories to tell us. They have a huge diversity of sizes, and you can discover so many interesting things in them. So with that, I'll end, and thank you for listening.
For discussion, ma'am, can you take a few questions?
Yes, of course. I know I've got. Yes.
Well, very nice talk. I have 3 questions. One is the figs are seasonal, right?
No,
They
they're
are not? Okay. None of them are seasonal or some of them are?
Okay, so if a wild fig species has to remain, some trees in the population have to have these microcosms at any one time of the year. So they are asynchronous across the population.
Okay.
So in January, you might have this tree, and later in February, another tree. So they have to keep cycling the wasps.
Okay. So that
And
was the
that's
reason I
why
asked.
fig trees are only tropical and subtropical. You cannot have figs in a temperate environment.
The second question I have is you were saying these ants are learning the behavior for the odor. Do you know anything about what is happening with their odor receptors during this learning process?
No, it's a bit difficult to study that, but that's a very good question. But we certainly know that they are learning the association.
The third question I have is there any role of microbes, for example, fungi?
That's something that we started but didn't quite finish. We know that the internal microcosm is sterile. We have taken swabs from inside, and we don't culture fungi. And the reason being that the fig itself is producing a lot of proteases and other antibiotic compounds. So what we have done, and we haven't published this work yet, so we collect wasps as they are coming in because they are like aerial plankton. They pick up spores, they pick up all sorts of things. We swab them before they enter the fig, and we can culture a lot of stuff from them. But once they are in, and if you you open up a fig under sterile conditions, you don't culture anything. So we don't know the sources of the antifungals and antibiotics, other than the literature says that there are a lot of proteases, like phycin is a known protease. Like papain in papaya, you have phycin in figs, but we haven't taken that project further. But that's an excellent question. Yeah.
I have 2 questions over here.
Yeah.
Very interesting talk. My first question is on the microcosm volume that you are talking about. Does it directly translate to the size of the fruit? Does it mean that the more pollinators enter in, or the more entries at the later stage leads to a bigger sized fruit that we eat? Does it mean so?
Absolutely. And maybe therefore more attractive to a disperser.
Okay. The
Yeah.
second question I had was regarding the male pollinators that you were talking about. I thought I understood that the males, they are produced inside the microcosm and they die within. So how they can be a pollinator? Or was it experimentally done?
Only the females are pollinators.
You were talking about male pollinators.
Males of the pollinator. The females, it's one species, right? The males are not the pollinators, the females are.
Okay. Thank you.
These moths are synthesizing phytohormones. This is what I mean.
The
This
wasp
is
could be, yeah. We know many insects, they
Oh.
produce, cytokinins. They produce,
But here in this case, they are producing phytohormones. And that's why this the synergy between figs and these moss, this overall system, I mean, these phytohormones, fantastic, huh?
it is fantastic
Ah.
because galls, see, these are galls which are normally parasitic. But here you have a mutualist that is producing
Hmm.
a gall.
Hmm.
So that's how the parasitism has turned into a mutualism.
Again, so second question. So when you were doing that sequence analysis, so you found those sequences there in the moss, which are capable of producing
So
phytohormones.
we are exploring bioinformatics is not yet done for our species. We have the full genome. In other insects, the genes have been found to produce plant hormones. Yeah, but we have to confirm it in ours.
Ma'am, very interesting talk. I wanted to ask, what is the relationship of the microcosm and the quality of the fig which we eat or the taste?
Okay, so the figs that we eat are parthenocarpic, meaning they are stimulated to produce the figs and the seeds without the wasps. So you can eat the figs very happily. They are perfectly vegetarian. Okay, but if you eat wild figs, then you will get the nematodes and they're very tasty. I can attest to that. Did that answer your question?
How does the microcosm affect the quality of the fig?
Well, as I said, it relates also to the question that was asked by that young lady over there. The more the pollinators, the more galls, the bigger the microcosm. And therefore, there are, the ripening process. So some figs, I don't know whether you all have done any agriculture here, but there are fruits that are climacteric and non-climacteric. Have you heard that? So climacteric ones are maybe like banana. They go off very fast. The ripening process is very quick. Yeah, because there is a spike of ethylene. So if you want to ripen something, you spike ethylene, you ripen it. So people have studied in the figs. Our model species is climacteric. The moment the wasps are released, ethylene spikes, and within 24 hours, the fig is ripe to be eaten. Okay? There are others like Ficus bengalensis, the Bengal fig. That is non-climacteric. So the figs will remain on the tree for a very, very long time without really rotting or ripening. So even within figs, there is a variation. And if you follow that logic, you can then make predictions as to what should be the insides, the number of occupants. You can make predictions about it. Yeah?
Ma'am, here. So actually, at NIB, we have this ficus racemosus, like we have almost one plant at one side and other side. We can see they seems like they're completely synchronous. The fruiting pattern and you can see the figs, they're synchronous. Almost.
Within the tree, they will be synchronous.
Yes. No, within these locations, one at this side and one at other side.
Okay.
The same question I was about to ask, like seasonal. So you have a 24 hours of time. Within that, I don't think I saw somewhere near around 3 to 5 kilometers. There is another fig plant, Ficus racemosus. How far these guys travel and where do they? I mean, they have to stay somewhere within these other periods. Like, is it really possible in that
They
way?
have to, and they will do it. You can do an aerial survey. We have done that in Bangalore. So we have mapped most of the Ficus racemosa in Bangalore, and we have calculated distances and stuff.
Unless I have a question, you have a crosstalk between like Ficus bengalensis versus racemosus?
No.
No?
No
Okay.
crosstalk.
And the next question, so when we see when the fig is really kind of green, the local microorganism content, such as insects, you take all of them, when we open it, try to eat, always it will be very less. When it becomes like fruit and little bigger, we can see tons of insects. What is the reason and how do they coexist together at the later time points?
What type of insects? Are you talking wasps? Are you talking?
We can see.
It can be dipterans. I haven't talked about the dipterans. There are dipterans that enter some fig syconia. We don't have dipteran infestation in our population, but there are other populations where dipterans could also enter.
I mean, do the fig plant has any kind of advantage during the evolution because of these microcosm within the fruit, other than pollination? Because you're having other varieties of insect, whole world is there.
For the fig, the pollinator and the fig tree is one mutualistic loop. Okay? Pollinators can sometimes get greedy. What do I mean by that? We are all greedy, right? They might try to overexploit, lay many more eggs and do less pollination. The fig is sensitive to that and it will drop those microcosms. There are beautiful experiments that I haven't showed you that the fig is sensitive because it doesn't want to have a microcosm in which there are more pollinators and few seeds. So it wants to balance that also. Okay? The parasitoids that prey upon the pollinators and keep the pollinator populations down are an indirect mutualist. They are not direct mutualists, indirect. So we have also demonstrated that the parasitoids are like any carnivore in any ecosystem, they are keeping the levels of the herbivores also under control. So though they are not directly involved in the mutualism, they are coming in through an indirect loop.
Yeah. The final question, during this entire process, does the wasp lose any kind of fitness?
How would the wasp lose?
Because the female pollinators, basically they go inside through a very tiny nostril. After that, you have to make another way to grill all the stuff to come out of it.
Their offspring are doing it, not the same ones.
They'll die over there.
The females that come in, they die after laying their eggs and pollinating.
Okay.
In a few hours, they are dead.
Okay, got it.
Got it?
This very nice job. What is that size? What's the number of female pollinators can go inside? Which is how it's the fig controlling it, or
the ostiole? I haven't talked about the ostiole at all. The ostiole is a fantastic valve, kind of like a valve. If there are no pollinators that have come within a certain time, the ostiole will remain open for longer to allow pollinators to come in. Once a certain number of pollinators have entered, the valve shuts. So there is a regulation.
So in case of endangered species, so when there's fertilization, the sperm, the moment it enters, the luna closes. So it is something like that what you're saying.
There is a regulation
Controlled by the fig.
by the fig. The fig is controlled.
The parasite will always act which is interested or only of the fig can do. The parasite.
It has to find, I didn't show you that. Can I just show one
Yeah.
slide? Because I ran out of time. These are parasitoids. They leave chemical footprints on the surface. So even if you, and we have got GCMS traces, so we know exactly because the wasps have tarsal glands, and when they walk, they leave footprints. So if there is another parasitoid that comes in, it knows which one has been there before. They leave there, and they need to know this because we haven't quite discovered that part of it. Because when they are poking with their ovipositor, they need to know whether there are any, because even the parasitoid offspring can only mate inside. So if there are no mates inside for the parasitoid, that's it.
Okay, thank you.
So it's complicated.
Okay, so any more question from the students? Okay, then I'll just have one question from my side. So, ma'am, how do we use this information of the relationship of the wasp and the fig in terms of natural farming? Let's say when this fig, let's say this, this commercial farming was not there. There was this parthenocarpy base without any wasp, clean fruits was not there. How farmers used to use this information? What is the best time to pick up a fig?
Okay, so this parthenocarpy is a very ancient thing. Even in the tombs in Egypt, if you go to the pyramids in Egypt, you will find hieroglyphics where they depict parthenocarpy. There were specialized fig growers that knew how to, produce parthenocarpic figs. But in the Mediterranean region, you have the species of fig that is present there, which I haven't talked about here. What we have here is a monoecious fig. What does that mean? That every microcosm has male and female flowers in the same one. There are other species, like the ones in the Mediterranean region, where you have males and females. The wasps are only breeding in the male trees, and it's the females in which the seed figs are produced. So the farmers have to ensure male and female, the wasps have to cycle from the males to the females. So it's a much more complicated. But in the Mediterranean, you could have figs with some wasps inside.
Okay, thank you, ma'am, for really fascinating stories. And let's have a big round of applause for this fascinating story again, please. So now I will invite our director, ma'am, to felicitate our today's distinguished speaker. Please, ma'am.
Oh, absolutely wonderful. Thank you so much. Wow, lovely. Thank you. Oh, nice.
Now I'll invite our colleague Shashi to have a vote of thanks.
Sorry, I went over time.
Good afternoon, everyone. On the behalf of Scientific Event Organizing Committee, I like to take an opportunity to extend our gratitude to all who made this event possible. First, we are very grateful to our director for the continuous encouragement and support in organizing such events, scientific events. And first and foremost, I extend our heartfelt gratitude to Professor Borges for delivering such an insightful lecture, which is a very much rich perspective on ecological complexity and natural ecosystem. Really, thank you, ma'am. It's such a nice talk. I also sincerely thanks to all the faculty members, students, fellows who contributed their enthusiastically participated in this talk and engaging in a discussion. We also would like to thank technical and administrative teams who seamlessly coordinated for this efforts event. And finally, thank you for all for attending this talk. Thank you. Please come in front of the podium for taking a group photo. Thank you.
Oh, right.